Hello, everyone. Today, we're here to explore the really deceptively simple question: is blood sterile? And the ways in which my team reached a conclusion about this. So, for hundreds of years now, scientists have used various methods to describe and measure components of blood. Now, this sketch was done in the 1700s by one of the first microscopists, Anthony van Leeuwenhoek, depicting red blood cells. But we now know a lot more about the complexity of blood as a tissue. Within this molecular soup of salts, proteins, and other organic compounds, blood contains numerous host cell types that function in oxygen and nutrient transport and immune function. However, blood is traditionally thought to be sterile, that is, devoid of microbes, that is, except during infections, right, such as those leading to sepsis. And here we see one of the first reports about microbial cultures from the blood of diseased individuals around the end of the 19th century, right? But apart from clear cases of severe disease like sepsis, live microbes have also been described in the blood of individuals with little or no clinical symptoms of infection. This phenomenon is called bacteremia, and it can be caused by a weakened immune system or even a simple act of over-vigorous brushing of a teeth when you have gum disease, for example. Since the 1960s, there have even been reports of live bacteria in the blood of overtly non-diseased individuals. So these are healthy individuals with no clinical symptoms. And these observations lead to some important questions. Now, is it common for healthy individuals to carry microbes in the bloodstream? And when this happens, do healthy individuals harbor standalone blood microbes or a multi-species blood microbiome? Now, for the sake of clarity, let's define what a microbiome is, right? So the mere presence of bacteria, fungi, or viruses alone is not sufficient in our view to define a microbiome. Instead, a microbiome refers to that community of microbes where members of this community are alive, they can replace themselves, and different species can cooperate or antagonize each other. Another concept related to the microbiome is the idea of core species, which are microbes that are common to most, if not all, individuals at a given body site. For example, Cutibacterium magnes is a ubiquitous microbe on skin, right? So microbes have been described and characterized across a range of body sites: the oral cavities, the skin, the gut. But a controversy remains, right? Is there a blood microbiome? So we felt the need to rigorously investigate the presence or absence of the blood microbiome, given the growing number of recent papers using NGS techniques to claim one, right? And to this, we leveraged the Singapore SG10K health data set, which comprised of over 9,000 healthy individuals. We did whole genome shotgun sequences from the blood of these healthy volunteers across different ethnic groups. And with a study of this scale, coupled with a rich experimental and host metadata, we then tried to answer the following questions. The first, what microbial signatures can be detected in blood? Is there a blood microbiome? And if so, where do blood microbes come from, possibly? So now, when we analyzed the data, we found that the number of microbial reads were many magnitudes lower than human reads. So this is a low biomass situation, right? So the relatively low microbial signals in blood, coupled with various handling, experimental, and computational artifacts, means that there's a very low signal-to-noise ratio when you're trying to measure the blood microbiome with NGS techniques. Now, computational errors, or the false positive assignment of reads to a non-existing microbial species, can in part be addressed by performing stringent quality control, such as removing samples with too few microbial reads and removing classifications that are microbial signals that are too low in abundance. But we also accounted for experimental artifacts, which are contaminants introduced by the library preparation process, or the ketome, which some of you might be familiar with now. And we identified the ketome using a batch analysis and correlation strategy. Now, this decontamination strategy is only made possible because our dataset comprised of 6 different cohorts with rich metadata about the types of kits used and even the lot numbers of these kits. Now, the heterogeneity of these cohorts It's a challenge for analysis, but we turn it to an advantage by using batch-specific information to identify contaminants. And so having the assumption that microbial species that are present only in one cohort are likely not real signatures. So as an illustration of how this process works, we identified microbes which were differentially prevalent across different library kit batches. So this Spingobium YG1 species is a soil bacterium that's enriched in some flow cell lot numbers but absent in others, as you can see from this chart. And this makes it highly suspicious. But members of this genus were previously detected in the blood of healthy controls in other studies, right, as well. So, but our data set then suggests that this species is likely a false positive, at least in our data set. Now, we extended our search for contaminants by reasoning that other microbial species are also considered contaminants if their abundances were highly correlated with any contaminant within the same batch. So this heat map of Spearman's row shows that the abundance of most species of the genera Spingobium, Spingomonas, and Methylobacterium, which are environmental species, microbes, tend to be highly positively correlated with each other, and this makes them more likely contaminants from the same source. And we can then remove them and account for them in our analysis. We then ask the question if after removing all these environmental and kit-based contaminants, whether we have enriched for microbes known to colonize human organs. Now, after accounting for computational and experimental artifacts, we ended up with a list of over 100 microbial species found across our population of healthy blood samples. And reassuringly, when we check the likely origins of these 117 species to a previously curated list of kit contaminants, a microbiome database, and a literature search, most of the microbes that survived our filters were not known to be common contaminants, and they were human-associated microbes. So this suggests that we have increased the signal over noise for detection of blood microbial signatures in our study. We then asked the question where these microbial signatures originated from. It's important to note that these blood microbes are very sparsely detected in our data set, and signals are only present in 16% of our individuals. Most individuals, in fact, only showed signal from a single species, and we could not then find a core or common blood microbe that's shared amongst most individuals. In fact, the most common blood microbial signal was found in fewer than 5% of individuals, and it belonged to Cutibacterium magnes, which is a common microbe on skin. Now, this could be a real biological signal, or it could be an artifact of the blood drawing process. We can't exclude that possibility. But I think what's more interesting is that we also found other species belonging to the gut and the genitourinary tract, such as Bifidobacterium and F. virginiae species in the bloodstream of some individuals. Now, overall, we observed that most blood-borne microbes or their signatures were associated with various body sites or organs. Many blood microbial signatures can, in fact, be associated with the gut or the oral mucosae, and some in the genitourinary tract. And this would suggest then a model where we have sporadic or transient translocation of some microbes or their DNA from these disparate body sites into the bloodstream. We also investigated if these microbial signatures in blood could come from actively replicating bacteria. So if bacteria are actively replicating their DNA, there will be this telltale gradient of reads, as you can see here, originating from the origin of replication, here shown in red, and decreasing to the terminus of replication, shown in blue. And here is a set of non-contaminant species in which we saw evidence of DNA replication. These microbes come from diverse body sites. So F. nucleatum is a known oral and gut microbe. F. virginiae colonizes the genitourinary tract. And A. baumannii is found on skin or in respiratory secretions. Now, this points to a model where live microbes, or at least the DNA originating from live microbes, can occasionally transit from different organs into the bloodstream. But in contrast, when we performed the same analysis on a set of contaminant taxa, we did not find statistically significant evidence for DNA replication for contaminants. Now, given the DNA signatures of replicating bacteria, we investigated whether or not microbe-microbe interactions could be detected in the blood of healthy individuals. And for this, we just computed pairwise correlations between species, where you have positive values would suggests a co-occurrence or cooperative effect, and negative correlations indicate some sort of competitive or mutual exclusion effect. Now, when we visualize the correlations of the 117 blood microbial species using network graphs, we could not detect any strong community co-occurrence or mutual exclusion effects. And in fact, if you look at the correlation coefficient, most associations are very sparse and weak, even across a very lenient range of correlations. Now, you contrast this with a network graph from a gut or oral microbiome, and you can see many more stronger interactions between various species in these other microbiomes. I think this then drives home the point that microbes in blood lack the hallmarks of an actual microbial community, because they exist probably in isolation, and they occur transiently as well. So in conclusion, we began today's talk with a simple question: is blood sterile? And we now know that the answer is a lot more nuanced than that. So for the practical purposes of blood donations with proper regulation, blood is sterile, of course, and is safe for use, but blood is not completely devoid of microbial signatures. Even so, we find no evidence to support the model of a multi-species blood microbiome with associated microbe-microbe interactions. Instead, we think that sporadic and transient translocation of commensals from other body sites into the bloodstream is more likely in this model. So this raises some more interesting questions we can discuss later on in Q&A. Are microbial signatures from viable cells, or are they from cell-free DNA? Are there experiments where we can show evidence of actual microbial translocation between body sites into the blood? And are there diseases, or in the case of age in elderly individuals, is there an increased frequency of this transient translocation of microbes into the blood? Now, I'd like to thank my team, especially the first author of this study, Cedric Tan, a student from our lab, who did the analysis. And if you're interested in the technical details and our conclusions, this paper has also been published in Nature Microbiology this year. And I thank you all for your attention. Thank you.